Welcome to the Everything Epigenetics podcast, where we discuss DNA regulation and the insights it can tell you about your health. I'm Hannah Wett, and I'm the founder of Everything Epigenetics. Today, I have an awesome guest for you. His name is Dr. Gabriel Fries. First, we hop right into it. You all will get a nice introduction and hear from him about his personal journey into his psychiatric research, focusing on molecular biology and epigenetics. We then hop right into epigenetics and psychiatry. So what are the epigenetic mechanisms mediating the interaction between genes, environments, and playing a key role in psychiatric disorders like bipolar disorder and suicide risk specifically? We also talk about aging in psychiatric disorders. So how psychiatric disorders are really linked to accelerated aging and higher rates of age-related diseases. His research really highlights that bipolar disordered patients, especially those with suicide history, have older epigenetic ages. We also get into lithium in aging. So lithium may have shown some anti-aging effects such as telomere length elongation in bipolar patients. And then we dive into ongoing research. Dr. Fries is specifically looking at markers and investigating drugs that could possibly prevent or reverse epigenetic aging. We finish up talking about future directions and where he's taking his interventional studies in the future with some in vitro drug trials, focusing on reversing those epigenetic biological age clocks. We then end by speaking about his challenges and future work he's excited to continue on. So very briefly about my guest today, Dr. Fries is an assistant professor in the Department of Psychiatric and Behavioral Sciences and a translational researcher in the field of biological psychiatry. Again, he focuses on the epigenetic bases of mood disorders, particularly bipolar disorder, suicide, and mechanisms of stress. All right. He also collaborates with basic and clinical investigators to search for the genetic and epigenetic underpinning of severe mental illnesses. Now, he's received several grants. He's a principal investigator, and he's currently funded by the National Institute of Mental Health and is running the UTHealth Department of Psychiatry and Behavioral Sciences. And now for my guest, Dr. Gabriel Fries. Welcome to the Everything Epigenetics podcast, Dr. Fries. I'm excited to have you today.
Thank you. I'm excited as well. Thanks for the invite.
Yeah, and we'll start off with just some general knowledge about your background. You know, I'm always so curious to hear how people end up in their particular field, whether they took a very traditional path or maybe not so traditional. So your history in particular is extremely interesting to me because of the subject we're going to be talking about today. So can you just give us a little bit of background on your journey and how you ended up, you know, doing what you're currently studying and looking at?
Mm-hmm. Sure. Well, it's supposed to be a simple question, but sometimes it isn't. I know how to answer that. But I've always known that I wanted to do scientific research and medical research in general. And when I started my undergrad, my college experience, I started to try out a couple of topics. You know, I immediately entered into a research lab, but then, you know, fell in love with biological psychiatry, as we call, like this idea that we can contribute to the psychiatric research by providing some biological insights and some molecular underpinnings to these disorders, which normally are not seen or are not necessarily seen as medical diseases in that sense, right? It's easy for someone to understand cardiovascular disease as something that is related to molecules and all that. But when it comes to psychiatric disorders, there sometimes are some limitations and some restrictions as to, okay, that may not be the case. But in fact, I truly believe that it is. And we've been doing this work for many years now. So I did my master's and my PhD specifically in biochemistry. So I really wanted to get into the molecular level of everything with a focus on psychiatric disorders. Came to the US. So I'm originally from Brazil. So I came to the US to do a postdoc 8 years ago. Here in Houston, and the topics come out of each other, right? So we started working with epigenetics in general. That has led us to study epigenetic aging and aging in general. And one thing keeps leading to one another, but I still am very passionate about the field and excited with what it can lead to and the contributions that we can make in the future.
Yeah, for sure. And I think it's safe to say you're an expert in the epigenetic basis of mood disorders and psychiatry. You perfectly outlined what we're going to be talking about today. So that kind of leads me directly into my next point, which is great. I want to really dig into this though, because you have all of these really great papers. I think it's better if we start big picture, like you mentioned, and just talk about epigenetics as a subject. So, why is it important to study this, more in particular, the psychiatry in relationship to epigenetics?
Yeah, so epigenetics is a really interesting um, route to study psychiatry for a couple of reasons. So just for the sake of introduction, right? So we're talking about mechanisms that can affect gene expression in response not only to our genotype or to our genetic background, but also to the environment, and that fits perfectly with what we believe um, is implicated in mental health or psychiatric diseases, right? So they are all known to be multifactorial diseases, meaning that one needs a risk genotype to interact with environmental triggers or things that happen throughout life. And so it makes sense to think that epigenetic mechanisms such as DNA methylation, non-coding RNAs, or histone modifications are mediating this interaction between genes and environment. So we've known for many years that psychiatric disorders are heritable, meaning that they are clustered in families. Offspring of individuals with diagnosis tend to have a higher risk of developing these disorders. So there's this genetic component that right now cannot be fully explained by just looking at the DNA sequence. And that's where epigenetics and our work comes into play, is to try to complete that whole picture, right? To paint the whole picture, in addition to ongoing genetic studies per se that are actually looking at variants and mutations and things like that.
Mm-hmm. Sure. Yeah. I just think it's so interesting. I think if I were to go back through school, I definitely took a very traditional route of the general biology degree path. I would go back and do more of like the bioinformatics or definitely psychology. I remember I took like psychology 101 and it was just fascinating. I've always been interested in that. So again, that's why I was so excited to have you. So now we'll go a little bit deeper, but talk, I guess, more general in terms of epigenetics and then aging and psychiatry, right? So we get to this idea of epigenetics. People usually tie it into aging. So, how can we look at these factors and maybe mitigate or reverse some of them as well? So, when you're looking at psychiatric disorders and epigenetic aging from a broad perspective, what are you really finding? And we can go through some of your papers later as well, but you can even go a little bit into the specifics too.
Yeah. So the big picture here, or the basis of all of these aging epigenetics and psychiatry studies that we do, are based on the fact that most of these disorders are associated with a premature mortality and or a higher incidence of age-related diseases and cardiovascular disease, diabetes, dementia, um, or a faster clinical cognitive decline over time. So there were these clinical suggestions now that an acceleration of the aging process may be taking place in some patients with psychiatric disorders. And in a sort of independent line of research, there have been many studies proposing that the process of aging includes epigenetic mechanisms, as you know, and I'm sure you've been talking about this over and over with different guests here. So this is when these things came into play, right? We thought a couple of years ago, and we were certainly not the first to do this. We thought, well, maybe epigenetic mechanisms may be underlying these, I think as these findings that people were having, that there's an accelerated of the aging process in these diseases. One sort of low-hanging fruit to study this is using DNA methylation levels to estimate these so-called epigenetic clocks or markers of pace of aging, and these things that you guys are very well familiar with. And so we started doing that with a lot of the samples that we have. And in our lab, we're particularly interested in bipolar disorder and suicide. Those have been the main focus, even though we've worked, in collaboration with other groups that have included samples from other diseases. And in general, we do see a pattern that's similar to what we would expect. In a way, we see higher or older epigenetic ages or age estimates in patients with these diagnoses or individuals with these diagnoses compared to non-psychiatric controls. And a couple of things. That's not seen in every single patient, so that's an important thing to establish. No, it's not a deterministic association, and we see some clinical associations that may be of interest. So, for example, in the context of bipolar disorder, in general, you see that the diagnosis is associated with the higher acceleration of the epigenetic aging process, but within individuals, those that have a history of a previous suicide attempt may have an even higher acceleration of the epigenetic aging process. So we see these nuances and differences even within the large diagnostic clusters that we talk about.
Gotcha. Yeah. And why do you think that is? Is that what your main questions are now, is trying to dive into that a little bit further? Because I have here, as we're kind of going through, our agenda, you look at all of these different disorders, right? You mentioned mostly the bipolar and suicidal, but I have, schizophrenia, different anxiety disorders, substance use disorder as well. So it's you're really trying to find those different subgroups and all of these factors which may be affecting those clocks and what that means.
Yeah, I mean that's the question now. Now is to try to find the drivers of these findings. Now that we're just seeing, I think that to some extent we're just finding the final outcome, if you will, but we're not getting a sense of the process and what's necessarily leading up to those alterations that we see. Most of our studies so far have been in cross-sectional studies, so these things need to be seen with a grain of salt. We're looking at that one time point in the life of the patient. Even though we discuss it as an acceleration process, really when it comes down to it, since we're not following up these patients with these markers, we can't really argue that there's an acceleration. What we see is that something that happened up until that point has led them to have older epigenetic ages. And we, of course, extrapolate and generalize those findings. But I always like to think that, we need to be a little bit cautious of that. Even in the limited context of a cross-sectional study, you can make inferences, or at least look for correlations and associations with clinical factors and other things that can maybe generate hypotheses for future studies. And so we see, for example, that within individuals with bipolar disorder, those that have a older epigenetic age compared to their chronological age, they tend to have worse cognitive dysfunction, if you will. Now, so we see this logical, but yet for the first time, actually empirically shown association between the aging process and its acceleration with higher degree of cognitive dysfunction or cognitive impairment, however you like to call that. Like I said, we see an association with history of suicide attempt, which is an interesting finding as well, to think that this one possibly very traumatizing event in the life of a person can have an impact in your biological clock. In fact, there's a lot of evidence suggesting that survivors of a previous suicide attempt also die earlier in a way, and that cannot be fully explained by suicide itself. So that it makes sense that maybe that aging process accelerates after that. I think that also overlaps a lot with the PTSD literature that I'm not, a part of, but I follow, in the sense that these individuals also seem to have accelerated epigenetic aging. So there's a lot of discussion about the importance of these traumatic, stressful events leading up to those. So yeah, this is there's a lot to explore there in terms of the causes. But since we're seeing these associations with potential consequences, now, which serves as the basis for most of these studies, I think that's interesting. I think that's worth pursuing the mechanisms that are leading up to those alterations, so that we can start to target them, in fact, right? So, of course, it's only a hypothesis at this point, but what if we can somehow prove that it's through these accelerated epigenetic aging processes that a lot of these age-related consequences end up taking place in patients? What if we then can use some of the findings that we have to propose ways to target the aging, prevent those consequences, and ultimately increase lifespan and promote healthy aging in patients? I mean, this is the significance, at least, that we try to focus on when doing these types of studies.
Exactly. And it seems like everyone I'm interviewing and all of my guests who are phenomenal, they all have that same goal eventually, right? They may be studying different subjects or different diseases or disorders, but yeah, it all comes back to, hey, if we can identify this, take more of a preventative approach, then we can get a better outcome. So I just think that's really the beauty in looking at these markers. And I know you mentioned you're basically getting the data from one time point. Are you able to follow up with a lot of those people from the study? Are you able to get any longitudinal analysis? Is that of any interest?
It is definitely of interest. Our group here in Houston, we have some patients that come. It really depends on the study that they're enrolled for. But we haven't been able to run these analyses yet. So in fact, this is one of the things that I'm doing right now is I'm trying to reactivate this discussion in our department to start recruiting those people again. Because of course they're all volunteers. It's a hard process, right? Any longitudinal study is tricky. Or even find collaborations. Maybe a listener of the podcast who has similar samples that would be interested in letting us help with the process of measuring some of these markers. That would be wonderful. Because I think that's a very logical next step in our research and something that we want to pursue. I mean, like I said, we are extrapolating a lot of things from these cross-sectional findings, and that's great, albeit limited. But the next step is exactly to see what comes first, right? So we have some unpublished, very preliminary evidence that maybe some of these accelerated epigenetic aging markers, they are associated, for example, with metabolic dysfunction or inflammation. So, not going to talk much about it, but we do see these things. And now the question is, what comes first, right? Is it is it the case that these diseases, these disorders are leading to some of these somatic medical changes that ultimately lead to the accelerated aging process, also mediated by the environment and all that, which makes a lot of sense. But is it the opposite, right? Is it maybe an epigenetic aging process that's already accelerated, that maybe to some extent is already being dictated by the genetic profile of these individuals, that then contributes to a lot of these metabolic, inflammatory, and many other alterations that will end up activating and leading up to the higher risk for age-related diseases, dementia, et cetera, et cetera. I think that this is an interesting discussion that will probably only be explained when we really look at large groups of people over time, right? Longitudinally.
Yeah. It's an interesting way to look at it. But as more of the data and research comes out, we'll be able to answer some of those questions, which will be great.
Yeah.
So I'm going to keep digging a little bit deeper here. And, this comes up, I would say, quite frequently, but I really want your take on this as well. There are a lot of epigenetic age clocks out there, and they're all great. I think there's ways you can use each of them, and they each have their own application. In your most recent work, however, you note that one of the strongest associations with mental illnesses have actually been in those newer second generation base clocks. And those are going to be the biological age ones. We've defined that in previous shows before, rather than those chronological ones. So these second generation ones, the PhenoAge based on clinical measures of phenotypic age. And then we have the GrimAge based on DNA methylation surrogate plasma proteins linked to different outcomes. So can you just speculate on that? Why do you think that is?
Yeah, I think it has to do with the way by which they were developed, which echoes what you just said, right? The first generation clocks, as we call them, Horvath, the Hannum, they had the sole purpose of predicting chronological age, or the time since you were born. Um, and again, how great it is to have a marker like that for many different reasons, for forensics even, a lot of different applications. But we know that chronological age is a very poor predictor of age related conditions. So just by having something that predicts your chronological age, when you look at the clinical significance of that, is probably just as limited as the chronological age as a variable itself, right? I mean, again, we can extrapolate, we can compare that to the chronological age and see if it's older, younger, and make estimations out of that. But that was not the point of those original clocks. Now, like you said, PhenoAge, GrimAge, these second generation clocks, they were the idea was exactly the next step was to not focus on time since birth, rather on actual biological markers of aging or things that were related to the process of aging. One of them being the time until death, right? Time to death, which makes a lot of sense for me. I mean, this is the ultimate consequence of the process of aging, which is the GrimAge itself. So it's really not a surprise for me when we published a lot with the first generation clocks. When we started calculating the second generation clocks, that's when most of the very interesting findings started to come to light. Because of that, I think that we're finally capturing meaningful biological aging processes. And of course, we nowadays have even more sophisticated ways to estimate that, which is these third generation clocks that
Mhm.
you're well aware of.
Yeah, I think we'll start to see the research shift that way. I at least hope, right? It's nice if you get first gen, second gen, you can maybe all of them in there, so you may be able to see correlations and trends. But I really hope to start to see that shift to more of those second generation clocks, because sometimes I really don't think that people are aware of those different generations, right? They hear biological age and get so excited. So I just always like to point that out and note that difference. But
Yeah.
you're exactly right. What about the DunedinPACE? I heard you throw it in there at the beginning. So have you looked at the implications of psychiatric disorders and all of your work with the pace of aging metric?
Yeah, so I'm going to disappoint with my answer here because we started looking into that. So we used a lot of the data sets that we've worked to up to now and generated DunedinPACE from them. I mean, thankfully, the codes and algorithm are open for researchers. So kudos to the group of Terry Moffat and all that for making this available. And we're starting some analysis on that. I can see that they're very excited, but there's we haven't published anything. It's really what we're doing right now is trying to follow up with these new markers, which I'm very excited about. I mean, I think that being able to capture pace of aging, for one thing, is a huge advantage in the case of our research questions over the existing clocks that are out there. Um, Not to say that the clocks are limited in a way, but they're limited for what we're currently using them for in our research, right? So we make a lot of extrapolations about the acceleration of the aging, and we make a lot of assumptions that really are about pace of aging, if you think about that. Um, and so that's why I'm excited to actually have a marker that nowadays can directly assess that. Um, yeah, so it's ongoing. I hope to have some really cool things coming out soon.
Well, good. I like how you laid the groundwork. You said, "All right, you all will be disappointed, but here it is." Well, no, you're currently looking at it, right? And that's kind of where my mind went when you said, we're looking at people who had a suicide attempt and they're aging afterwards, right? Like my mind went to, oh wow, that's super interesting. I wonder how their Dunedin pace would play out after that, right? So
Yeah.
even furthermore, the longitudinal data of the Dunedin pace too, right before and after. So it's all super interesting, and I think we'll unlock some different levels as you start to
Yeah.
dig dig more into that.
Yeah, and the suicide is a obviously a super sad event, but it's a fascinating topic of research in that sense because you have different phenotypes as well. So all of this stuff I just shared with you that's published that's related to suicide attempt, but we're doing a couple of studies nowadays to see what happens during the suicide ideation, for example, um, and that's the type of marker I think DunedinPACE that would be really interesting to check because, for example, we're looking at patients during an acute suicide ideation event and after recovery from that episode. And so, how cool will it be to see whether the pace of aging changes as you recover from these very toxic brain events?
Yeah, yeah, no, that would be great. And I could just tell how passionate you are about everything you talk about. So I have to ask, what's your favorite subgroup to study, or where does your passion really lie? Do you have a favorite?
Um, well, I probably should say bipolar disorder, just because of the track record of it. I mean, I've been studying it since my master's degree back in the day when I was in Brazil, and this has led me to come here to continue working with bipolar disorder. It remains the main topic of our studies, even though we try to expand a little bit and see, other events and other subphenotypes in the context of bipolar. So that's when we talk about suicide attempt and ideation within patients with bipolar disorder and things like that. Um, yeah,
Gotcha.
it's just a topic that's of really great interest to me. I think that there's a lot of opportunities to help patients. Yeah, and I hope to be able to continue in the field.
Yeah, no, that's great. I had to ask because it's just radiating through you. So we appreciate all this great work you're doing. Um, we're going to keep digging into this further. So one other important point you mentioned in your work is that the features of this accelerated aging are not found in all patients, but there's with substantial heterogeneity. So even within the same clinical diagnosis, and you can explain what that all means. But I think this is profound, and I'd like to understand this a little bit more. So can you discuss why you see this and maybe what it means? Just a little bit of a review for our listeners.
Yeah. Well, I think it's not surprising that whenever we measure any sort of biomarker or biological alteration in these individuals, um, you never see the same pattern in all of them. Let's say compared to people that do not have the disorder. I mean, the same way that if you compare two people in front of you that have a diagnosis of bipolar disorder, let's say, they're going to be very different from one another, clinically speaking, right? They probably don't have the same combination of symptoms, and that comes, it comes with that discussion of how heterogeneous the diagnosis is. And it will think when you go to the biology, you see the same thing. So even if, as a group, the diagnosis is associated with a higher, process of aging, like we're saying, if you look at the individual data, you have patients that, of course, have very high, but you do have patients that have very similar epigenetic ages compared to controls. It's really an effect of the group, and when you apply a statistical method there, you see the difference, and that's what I mean when I say that it's not a process that takes place in every single patient, and I think that's important because it shows you that first of all, anti-aging um, interventions, for example, may not be useful for all patients, right? We may be focusing on one very particular subgroup within the diagnosis. Um, and on the contrary, there may be potentially more severe presentations where you would already see accelerated aging patterns in, let's say, very young people. Um, so it's interesting to see this heterogeneity because you also it opens up for a lot of other sort of clinical implications. If you think about treatment, for example, it's very common that even gold standard mood stabilizers, antipsychotics, antidepressants, they do not all patients respond to those drugs the same way. In fact, I mean, it's a very well known fact that there's, for example, lithium, only one third of patients will respond excellently, an excellent way to the drug. So you have this variability that may be explained and may be captured by some of these epigenetic markers that we're studying. For example, aging. No? Like lithium has anti-aging properties. We've seen this in vitro. But it may not be beneficial for patients that do not have an accelerated aging process in that sense. And maybe that overlaps with lithium response. If you, I mean, I'm just really coming up with hypotheses here that we have not tested, but it's one of the opportunities that finding these subgroups of patients may offer.
Sure, sure. And we'll no, that makes a lot of sense. So it'll be interesting to see the reasoning behind that as well. I'm gonna run with the lithium that you just brought up, and we'll switch to that subject. So this is gonna sound so silly. I'm gonna say it anyways. As I was looking through your research, putting the agenda together and everything, I was like, oh, this is just going to be such a great conversation. And I spend most of my time speaking with healthcare providers about aging and different interventions and, the studies of what's been published. And I'm like, I need to go back to that lithium study I always talk about with my providers and see what Dr. Fries may think about it. And then I was like, this is his work. I just didn't put two and two together, and I should have known. So it's a little bit of an embarrassing backstory, but here we are, and we're going to chat about it. So yes, you have that mini review, and it's called The Anti-Aging Effects of Lithium in Bipolar Disorder. So, I think, like you said, we've always known in vitro that it'll have a effect or something with aging. So can you describe what you found in this study? I think most people who, at least my healthcare providers who are listening, are going to be interested in this part.
Yeah, there's a vast literature on lithium showing how neuroprotective it can be, or how about the effects of like lithium on inducing um, anti-inflammatory processes or protecting, increasing cell resilience against stressors and things like that, oxidative stress, you name it. It's a well-known neuroprotective drug. At least preclinically speaking, that's very clear. What people have found, and we later replicated in our lab, is that lithium also interferes, maybe through all of those pathways in terms of oxidative stress and inflammation. It interferes with the telomere length, which is, as you know, very classic marker of aging. And in fact, there are a lot of studies even going deeper into the mechanisms by which it can take place. I mean, through GSK3 beta inhibition, you can activate a bunch of pathways that ultimately interfere with the telomerase enzyme that can lead to the extension of that portion of the chromosome. We found a couple of years ago, and we published this here in Houston, that if you treat cells from, in that case, it was lymphoblastoid cell line. So it's one cell line that was reprogrammed from lymphocytes from individuals with bipolar and controls. We found that in the cells from the patients, lithium was indeed able to expand and elongate the telomere length, showing, that providing some evidence of its anti-aging properties. However, we did not see a similar effect in the cells from the healthy controls. So in a way, it shows that, yes, lithium can have an anti-aging effect, but it may not have the same protective effect across all cell types, right? So it's still not a good idea to put lithium in our tap water or something like that, if you will. It's still something that can come with a lot of adverse side effects. It may be protecting in patients and in certain situations. But it does seem very convincing. Again, this was a replication of findings from many other groups showing that lithium can have that effect in addition to its neuroprotective effects.
Mm-hmm. Yeah, no, that's great to hear. I wanted to grab on to that snippet that you mentioned. So thank you for that. What about the future development of your work? What does all this great research that you're doing tell us about novel treatment strategies for these diseases? So maybe, they could be more specific based on the subgroup, or where do you think that will lead to?
Yeah, that's what we're tapping into right now in our work, is to, first of all, really understand what's driving these aging processes, and by doing that, identify ways to prevent that or even reverse, if you will, if that's at all possible. And trying to find what we can do to prevent that. So we have a study right now testing or screening a bunch of drugs that, for a couple of reasons, we believe may have anti-epigenetic aging effects. So we're working off of the hypothesis that, as you know, one of the main one of the strongest findings in the anti-aging literature is calorie restriction, right? So it's the thing that we don't want to do. But if you stop eating, reduce significantly without inducing malnourishment, seem to delay the process of aging. So in fact, there are some drugs nowadays that can mimic the molecular mechanisms of that calorie restriction without actually forcing you to make that calorie restriction. So we're testing them in the lab, and both in cells from healthy controls and from individuals with bipolar disorder, to see whether they can in fact prevent or modify that epigenetic aging process. We're comparing that to, for example, lithium, which is, like I just said, a drug with alleged anti-aging properties. And more than that, we're trying to see whether we can expand these results into the real the clinical, like real world setting, right? So maybe patients that respond to lithium are more likely to benefit from anti-aging drugs, or maybe individuals that have a high accelerated aging process at baseline are the ones that are more likely to benefit from a specific type of drug, so on and so forth. So this is where it comes into play, and I think that relates a lot to this idea of pharmacoepigenetics in a way that maybe based on these baseline epigenetic markers, be it epigenetic aging marker or specific genes that are methylated or groups of genes that are methylated demethylation, we can predict whether someone can respond well to a particular treatment or not.
Yeah, and that's fascinating. And is that are those studies ongoing right now? The ones that you mentioned where you're looking at the different therapies that mimic caloric restriction?
Yeah, exactly. We've gotten funding from the Milken Institute to do some of those experiments. And I think that the ultimate goal is to find drugs that can be either fully, newly developed or even repurposed, like drugs that we already know are safe for humans. I think those would be the ideal ones. And those are the ones that we're testing right now. And, based on our results in the laboratory setting, potentially propose some clinical trials, with maybe starting with healthy volunteers, non-psychiatric volunteers, and then maybe even patients, see if that can have a positive effect as an add-on or even as a standalone medication.
And Dr. Fries, I want to touch in on that point there. So I know there's this need in these studies to have more true interventional studies. So, with everything we were just talking about, are those going to be purely interventional trials? What are some things you're starting to look for in your lab?
Yeah, so far, they are interventional in the sense that we're testing their direct effect on these aging markers in an in vitro setting. So we're not testing them in patients, but that's ultimately the goal, right? So what we're doing right now is we're screening several different drugs that have this calorie restriction mechanism linked to them, and then we hope to find those that have the most promising results in the epigenetic aging realm, and then maybe propose them as clinical trial later on for both individuals with a particular diagnosis or even for the general population. Yeah, I think that's great. Of course, we know the need for that in this epigenetic world, I should say. Most people are excited about that, but they don't realize there's a lot of work that goes into it beforehand to actually get to that point. So with those interventional trials, or even maybe some things that you've done previously in your work, what are some of the challenges in your field that you think should be addressed? Or, as a researcher, what do you run into that may be a little bit frustrating? Mm-hmm. That's a good question. I think that we we lack replication of a lot of the findings that we end up having. So, for example, my own group, we've published quite a few papers trying to convince people of why epigenetic aging is important in the context of bipolar disorder, suicide, and all that. But I want to say that most of the studies have all been performed with the same data set, pretty much, right? Or the same group of people here in the Houston area. So I really want more groups to start testing them in their own cohorts. And I think we need that for the field in general. We need replication. We need large groups of people being tested for the same thing so we can see what are the findings that survive this, not this replication trial. I think that in the realm of epigenetic aging in psychiatry, we definitely need more longitudinal samples to be run. We are making a lot of inferences and assumption about acceleration, but at the same time, we're studying cross-sectional data. So it's really not the ideal data set to test this. So definitely longitudinal samples. By doing that, I hope that we can start exploring in more details this heterogeneity of patients. Like we talked before, it doesn't look like every single patient has an accelerated epigenetic aging process. And I think that one of the perspectives of the field is trying to understand why that is, what makes one individual more likely or at a higher risk for premature aging, and while others, don't show the same pattern. Maybe those that do have accelerated aging processes will benefit more from certain types of treatments like the ones that we were talking about, or any type of intervention that can have an anti-aging effect. Another maybe bottleneck here that we're trying to explore is trying to understand what drives these processes, right? What are the underlying molecular mechanisms that actually lead to accelerated aging processes in these patients? And maybe by knowing those, we can target them and ultimately prevent and or reverse epigenetic aging in individuals with bipolar disorder, with previous history of suicide attempt, and so on and so forth. And ultimately use that to propose novel treatments. And again, tying it back to what we just discussed about these new potential anti-aging drugs that can benefit patients in a lot of ways. And I think that's the ultimate goal of our research, is to help patients, help their families, help clinicians, and hopefully reduce morbidity, premature mortality, the incidence of aging-related diseases, so on and so forth.
Of course. I think those are all very valid points. And as I have more people on the podcast, I'm getting more of a consensus of those points as well. So it'll be interesting to see how we attack that. But again, with time, I think all of those will come, right? We're going to get larger training data sets. We're going to get larger validation data sets. We're going to get more information as to causation rather than correlation, right? And then, again, setting up these, I don't know the right word I'm looking for, but these fundamental, I should say, caloric restriction trials, things like that, and being able to use that as a proof of concept study and then follow up with that cohort, 10 years later down the line, 20 years later, it really sets the framework and the groundwork for more things like that. So all heading in the right direction. But I think it's very important that people are aware that it's not easy, right? There are some road bumps and things that we have to overcome. So we're getting really close here. One of the last questions, Dr. Fries. What's next for you? What are you excited about? I know you have a lot going on, some of those interventional trials, but is there like one thing that really sticks out?
Cool. I think I kind of mentioned some of those when I was talking about the perspectives of the field. I basically was listing the things that I expect to do in our group. So again, we're following up on those molecules in an in vitro sort of setting, but we work with a lot of clinicians here in the department that can help us with the next step, which is to actually do small clinical trials with individuals with bipolar disorder. I'm very excited with new collaborations with these larger samples that we can then test the same sort of questions that we have answers for in our own Houston sample, and see how those things behave in other cohorts, looking for longitudinal samples. And yeah, just collaborating with the field and contributing to this very exciting possibility, which is to maybe target aging, and by doing so, improve the lives of patients. So I guess that's what's hopefully next for our group and for the field as a whole.
Good. Well, I'm glad to hear that, and excited to follow you along in this journey, and look at all of the work you and your team are doing and continue to do. So one of the very last questions, this is more of a curveball. I always say it's very random and it wasn't on the agenda, but Dr. Fries, if you could be any animal in the world, what would you be and why?
Wow. Okay. Unexpected question. I think I want to be a dog. Maybe a lazy answer, but I'm a big dog person, and I cannot think of a better life than to be a dog in a family. In fact, my family just got a puppy, so we're all about that right now.
Very cute. What kind of dog, or what's its name?
So her name is Abby. She's an Old English sheepdog. She's adorable, and she lives the best life that one could have, right? So she sleeps, she eats, she gets treats, she plays. Why would I not want that life? So
I agree. I'm a big dog person too. I have a dog. She's a Bernese Mountain Dog, and her name's Evie. And I do look at her sometimes and just say, like, I just wish I could be her today. Like, just eat, get belly rubs and everything.
Yeah, can we switch lives for a day, and you stay here, I'll stay here, and you go write the grants for me at work? Yes.
There you go. Well, we've come to the end of this amazing podcast interview, and for anyone listening who wants to connect with you, Dr. Fries, where can they find you?
Yeah, so I'm on Twitter at Fries Gabriel, and you can also find me in the UTHealth, University of Texas Health Science Center in Houston Department of Psychiatry website. There you can find links to our projects and to our publications, and you can always send me an email with questions or requesting papers. I'm always happy to talk to people, not just researchers, but patients, family members. It would be a pleasure to share some of our work with you.
Definitely. And I'll put everything in the show notes. That way people can get in contact if they're wanting to. So thank you so much, everyone, for listening and joining us at the Everything Epigenetics podcast. Remember, you have control over your epigenetics, so tune in next time to learn more. Thanks a lot, Dr. Fries. I appreciate your time.
Thank you.